This is my fourth and penultimate lecture with you, so I'm very excited. Getting towards the end. This will not be a 3-hour long diatribe. It'll only be a 45-minute long diatribe. So we're going to talk about what happens once you've got through all of this bioinformatics business and you actually have a result. Okay? You have a list of peaks, or you have a list of genes, or you have a list of SNPs, or you have some list of things that your genomics experiment generated that you need to now consider. What the heck are you going to do with it? How many of you have been in this situation where, whether it's a microarray, it doesn't really matter, and you've got an Excel spreadsheet ranked from the top to the bottom, and you just looked at the top couple of genes in the list? Has anyone ever been in that situation before? Kind of seen something like that? Yeah. So we're going to talk about that and how to kind of go at that with a better, more complete perspective. So this is the preview. We're going to talk about 3 different ways to take a list of things, and I'm going to use differential expression again. It's the thing that I'm most familiar with as the context, but mentally you can search and replace that. Every time you hear me talk about differentially expressed genes, you can think about differentially marked chromatin sites, you can think about SNPs, you can think about genetics, you can think about methylation, you can think about phosphoproteins. It doesn't really matter. The idea is that in any kind of omics, high throughput profiling experiment, you have measured lots of things and looked for some difference between 2 groups, and you're now going to say these are the things that are now statistically significantly different. This is the thing that is the most statistically significantly different. This is the thing that is the least statistically significantly different. And what kind of inferences can I make that go beyond the individual element, whatever that is, beyond the individual SNP, beyond the individual gene, beyond the individual peak, to talk about more higher level biology that may be going on. Okay. So we're going to talk about 3 things: overrepresentation analysis, or ORA, functional class scoring, FCS, and pathway topology. Here I was obviously clever enough to give you some acronyms and also even tell you what they mean. So we'll go into each of these in detail. But first, a quick little tangent. Since you're all in this course, I don't really need to convince you of this. You're all interested in doing genomic sort of experiments and figuring out how to analyze those experiments. But I think it is useful to pause once in a while and remind ourselves, why are we doing them? Because after all, these are fishing expeditions, right? I mean, we're all critical thinking biologists. We all either write grants or review grants or write papers and review papers. Why would you ever fund someone to just go off and sequence thousands of genomes just to find out what's there, right? It's not hypothesis driven. It doesn't, how is that going to advance science? Well, the point, obviously, that we make is that these are opportunities to generate new hypotheses. We're not setting out to show that something is true. We don't sequence 1,000 new genomes to test a hypothesis. We do that to actually make new discoveries, right? So it's discovery research. It's hypothesis generation. Okay. And this is just a little cartoon that I like to use. Everyone's seen this cartoon. Best way to find your lost phone or your lost keys is by searching under the lamplight. So too, lots of biomedical research is relatively low resolution. This is where we're looking, but if the truth is over here, you're not going to find it. And so if we can look more broadly and at high resolution, maybe we can move that focus at least closer to where the truth is. And that's what we're trying to do. What can't we do with genomics? And the thing that we can't do with genomics very easily, because we're profiling broadly in an unbiased fashion, we don't conversely get to talk about causality, right? All of these are inherently observational studies from the perspective of the things that you're measuring. You may have done some sort of perturbation in the system, mutant versus wild type, drug treated versus untreated. That's a perturbation that you've introduced into the study, but now your observations are just that, completely unbiased observations. You take all comers. You're not looking at any one particular thing to make a hypothesis. So, for example, in GWAS, when you get a SNP that is a hit, that SNP is not necessarily the causal variant of the phenotype that you were analyzing, but it is a signal that, hey, there's stuff going on here. This is now a hypothesis that you might want to look into and follow up on. So, okay. We can't control for various unknowns and confounders. So again, we measure gene expression, but we don't, if we're not also measuring microRNA, we're not also measuring transcription factor activity, and we're not also measuring transcriptional rates, we make assumptions about, when the abundance goes up in a particular gene, that means that something at the promoter happened. But it may also be that something downstream of the promoter happened, that the metabolism of the RNA itself changed. Maybe microRNA that target that gene changed. So when we see these changes, again, they are correlations. They're things that look interesting and are associated, but we don't always get to reason about them causally. The old adage, correlation is not causation, and identifying causation requires perturbation, hypothesis-driven experimentation. So we don't get to do hypothesis-driven experimentation with genomics, but genomics should lead us to a hypothesis-driven experiment, right? The other point to make here, and I'm not trying to be preachy here, but very often grad students and postdocs that I interact with, they do some sort of genomics profiling experiment. They have their data. They've got great, it looks beautiful. There's heat maps and there's pathways. And they say, where can I publish this? And I ask them, well, what have you discovered? Oh, I've discovered this, I've discovered that. And I said, well, which of these things have you tested? Oh, well, I haven't tested anything. But look, I've got this, this pathway here is the most significant pathway that showed up in my enrichment test. This pathway must explain and it's, and you look at this reference and this reference, it all makes perfect sense. I said, well, but have you tested it? Is that actually the thing that is going on? If you perturb that pathway directly, do you now mitigate or somehow modulate the thing that you were doing in the first place? Oh, okay, I guess I should have to go do that. So genomics is hypothesis generation, discovery research, but still good science. You have to go back and actually validate and test it somehow. Because this association that you saw, this is another kind of a hidden Markov model. This is actually a Bayesian network. If it seems like an association between A and B it appears, then possibly A is causally affecting B. But it's just as easily possible that there's some other confounding thing, some other actor, C, that actually influences both A and B together. And once you know about C and can take C into account, that correlation goes away. That's not actually there. Okay, so that's why you have to, anytime we have these things, you have to test that, because if you test that hypothesis and it doesn't work out, it means there's some other explanation of why that association was there. Okay, so this is what we do. We do lots of genomics, and this is kind of an old slide. It's got genotyping and sequencing and structure and methylation and chromatin and proteomics and metabolomics and lipidomics and all these sort of different things that we can do. And how they're all interrelated to each other. Is a really cute sort of infographic. And I'm using this just as a placeholder for all the sorts of things that we can profile. So we can profile broadly tens of thousands of genes, hundreds of thousands of peaks, millions of SNPs. We can do lots of things. Okay? And we measure those signals, whatever they are, and we find hits. Great. Things that seem above and beyond, at some statistical threshold, interesting. Sorry, I said that in the wrong way. So we measure things, and sometimes we have enough signal to actually see it. So 20,000 some genes in the human genome, but in any given RNA-seq experiment, you're only going to see about half of them. Maybe 70% of those genes are actually abundant in any given cell, in any given tissue. So already you don't actually really get to see everything. You get to see some of it. And then some subset of that is actually statistically significant based on some statistic and correction from multiple testing. Then from there, there's this kind of translation step. Now for gene expression, the translation step is really easy. I'm talking about this transcript, therefore I'm talking about this gene. But for a SNP or for a peak, that translation step may be a little bit more difficult if the peak is based on a transcription factor binding at a promoter that is kind of well understood and you know what it's doing, then sure, it's really easy to say, aha, this transcription factor is associated with this gene, must be involved with the biology of that gene. It's easy to make that positional guilt by association, but that's not always the case. So Bill talked about SNPs and other sorts of variation. What is the consequence of that thing? Does that SNP actually change the way the protein functions? So these are inferences that you have to make to translate the thing that you measured into some kind of functional concept. Okay? And some of that can be automated, and some of that is just something that you decide. Well, if my peak is within 200 base pairs of a promoter, I'm going to associate that peak with that promoter and with that gene. But why is 200 the right number, and why is it not 500? And those end up being kind of arbitrary. Finally, then you have some biology that you can now think of, but it's really just again a sort of a gene list or a locus list. It's some list of things that have passed through this process. And in there, again, kind of anecdotally, this just has been my experience, this may or may not resonate with you. There's some things you expected to see, some genes that went up and down, like, oh, right, great, that makes perfect sense. In fact, it makes such perfect sense because maybe they were even positive controls. Maybe when you went into the experiment, there were things you knew that you should see, and if you didn't see them, you would be concerned. About positive controls, what's interesting is that, of course, when we're running a gel or doing any kind of wet bench experiment, we always have a positive control and we always have a negative control. In genomics, we tend to have technical positive and negative controls, spike-ins and input DNA control and things like that. But at the level of our results, we don't always think about, well, what is it that I really expected to see, and are there things that I expected to see that didn't show up? What is my false negative rate? What are things that should be in the result set but aren't? All right, so I always sort of challenge people to think about that. So there's the ones you expect to see, and so fine, great. I haven't learned anything new, but it makes me feel good to see that they're in the result set. And there's other things that make sense, the kind of blue dots here, things that you can, based on your knowledge of your scientific domain and maybe a little bit of searching through PubMed, it's very easy to rationalize why that particular hit, why that particular gene came up in your system. Then there's the ones that are clearly genes for which there is knowledge, whether you have it or whether just it's sort of out there in PubMed, but you really can't rationalize it. You can't make sense of it in the context of your experiment. That might actually be a good thing. That might be quite exciting that, oh, this gene now has a new role, this very sexy gene that people are interested in some other domain. I can now bring it to my domain and start talking about how blue toe disease is caused by mutations in Notch4 signaling pathway, and this is really great. So, okay. But then again, if they don't make sense, then you can't quite connect it, and then it's hard to imagine what that next experiment is going to be. Because remember, at the end of the day, we want to try to take this information and design the next experiment, the next hypothesis test that we're going to do in the lab to try to test some of these things. And then there's all the rest. The things that they don't have an annotation, nobody knows what they do, it's locus 1, 2, 3, 4, 5, 6. And that could be the most incredibly fascinating biology in the world, but you don't have an antibody, you don't have a clone, you don't have primers, you don't have reagents, you don't know whether it's, where it's expressed or what its what its role is in disease structure. So if you're going to chase after one of these things, again, highly novel, high risk, but potential high reward. Maybe that's a sort of thing that you want to chase after. But there's just always there's this kind of these decisions that you're going to make as a scientist to try to put these things together. And I call these individual gene-focused hypotheses, single gene hypotheses, right? And this a vast swath of biology is concerned with single gene hypothesis sort of experimentation. Can I explain this function of the biology in a very reductionist way by focusing in on what this one particular gene is doing in the context of its work? So that's great. You can do that. You can consider that. And then at the end of the day, what tends to happen is the green and blue ones are the ones that you keep mentally. They're the ones that you understood them or you were able to make sense of them, and they're lower risk. They're what you can convince a funding agency to continue to have you research on. And they're the things that get you move forward. And everything else, these oranges and black ones, they tend to just sort of fall away, right? There are other things that, yeah, maybe some other postdoc in a couple years might pick up the the dregs of this experiment, but I'm going to race off with a couple sure bets that are going to make sense. Okay? And I think that's a reasonable strategy. There's nothing wrong with that, but it's just a recognition that that is what's happened. Well, we're going to talk about what the sort of pathway approaches that we're going to talk about is employing bioinformatic approaches to try to make sense of these gene lists, whether you know anything about them or not, or whether you think you can find out something about them in PubMed, in a way that you can actually find new connections between things that make sense and things that don't make sense, and even using some methods that use some of the raw data by itself to try to pull some of these things of completely unknown function, to pull them together with things that you actually know something about. All right? Because it may be that this protein of unknown function and consequence shows up in your list because it's directly interacting with something that you do know a lot about. And now you do have an angle. Now you have an experiment that you can imagine to go do to test this guy. So I like to pick on the systems biology folks because they like to show me pictures like this and say how interesting it is that there's this big structure over here and it's tightly connected. And I'll be honest, in my day-to-day work, I actually make these things and show them to people. And we get really excited about, oh, look, MAP kinase is right there, and these are all the things it touches, and all those other things were also disproportionately dysregulated in a way that is interesting. So these things can be informative, but this is not a hypothesis. I can't test whether this graph is correct or true. Okay? But it's kind of it's a part of what's out there. What we can test are the kind of what I call the cell paper figure 7 cartoons, right? At the end of all the blots and gels and graphs, there's some cartoon that says, this is how I think the thing works, right? And it's arrows and nodes and activation and inhibition. Those are the hypothetical models, and we have to come up with those ourselves. There's none of the methods I'm going to tell you about, no methods that have ever been made or are ever going to be made in the near future can do a good job of coming up with these models purely based on the data, on that cartoon of how the thing works. Okay? Lots of methods that can come up with these things, but this is not a mechanistic model. This is not a testable mechanistic hypothesis, whereas those figure 7 cartoons are. Okay? Now that said, there are ways that can, well, we're going to get to that. There's something in between. There's stuff in between here and here that I think computers and data can get to, but we're not there yet. So anyway, we devise these ourselves through some creative synthesis of the results and domain knowledge, either in your own head or that you find. And we have a preference. We prefer hypotheses that are testable and experimentally designable, things that we can actually test. Right? Because we can talk all day about wild hypotheses that may or may not be true, but if they're not testable, then we're kind of wasting our time. So we actually have, because of the reductionist scientific method, we have a preference for certain kinds of hypotheses that are not this. Okay? And we have some reasonable expectation of truth. This is the kind of the bar that you can have novel hypotheses that are wild, and that's fine, but the expectation of the community, not just the funding community, but your colleagues and your students, is that this isn't some wild goose chase, that there's some reasonable expectation of these things. Because the supporting data is overwhelmingly strong or obvious, that it expands upon known biology, and that you've actually done something to validate the thing that led it to you to that in the first place, through some other platform or some other repetition of the experiment, or seeing the same result in some new population. There are some statistical problems with this whole bottom-up process that we've been talking about. One is that when we start at the bottom and we do the statistics, whether it's chi-squared or ANOVA or the DEGsAnd EdgeR testing, we're testing each and every one of those independent entities separately. And we're saying that we're starting out by saying, well, they're probably all independent, when we know that they're certainly not independent. There's strong correlation. So GWAS SNP testing is a great example. Millions of SNPs, every single SNP is going to be tested independently of every other single one. And then because you've done 8 million, 10 million statistical tests, we're now going to have to multiply by 10 million to do the Bonferroni correction to talk about the statistical significance of each one of those SNPs. However, if we, instead of doing it SNP by SNP, did it haplotype by haplotype, there aren't 10 million haplotypes in a given population across these different things. You would greatly reduce the number of tests you were doing and probably increase the power significantly. And that's kind of what we're going to do when we do functional class scoring. Functional class scoring in pathways says, I'm going to take a group of genes that I think are going to be coordinately regulated or coordinately active. I'm going to test them together jointly instead of one by one. And maybe jointly there'll be some signal that is going to be stronger, statistically speaking, when I look across all of them than if I had looked one by one. Okay. We talk a lot about false discovery rate thresholds. I'm sure you've heard about false discovery rate. False discovery is a way of thinking about if I have 1 million p-values, I need to do something to address the fact that I've done 1 million tests, and I expect a p-value of 1 in a million approximately once every 1 million times if everything is independent. But we know they're not all independent, so we have to do something to adjust for that. And there's various ways to come up with the so-called false discovery rate, a transformation of the p-values into a different statistical number that tells you if I take all of the results that are at 10% FDR or better, 10% FDR or smaller FDR, then if I take that whole set of things that are at 10% FDR or better, then at most, the statistics tells me 10% of those will be false positives. Okay? Now, if at 10% FDR I only have 10 results, then that tells me that 1 out of 10 of them is probably not true. Hey, but the other 9 are. I don't get to know which one is actually false. The false one could be the very highest scoring one. Okay? So don't ever get fooled into think that at 10% FDR I can then take the top 90 and lose the last 10% and be safe, because you're not. The 10% of false ones are evenly distributed throughout that case. But why 10%? It's completely arbitrary. Why not 20%? Why not 1%? Why not just the top 50? No one of those thresholds is actually justifiable. It's just sort of your comfort risk zone, right? If you've got a result where the top 10 genes are at 10 to the minus 6th false discovery rate, then by all means you've got some statistical space, you've got some comfort zone to look a little deeper. Conversely, if your top 10 hits are bringing you into the kind of 20%, 30% FDR zone, then you're probably done. You can look at those top 10, but after that, you're really starting to get into noise, statistical noise. So FDR numbers are informative. They tell you about the relative risk, if you will, about continuing deeper into the list or stopping. But drawing a line somewhere and saying, all of these are good, real, ready to move forward, and all of these are not good, not real, throw them in the trash, doesn't make any sense. Speaking of which, what people tend to really do is they'll say, hey, here's all the genes that were at a 10% FDR or better in one experiment, and here's all the genes that were at a 10% FDR in some other experiment. Maybe it was another gene experiment, or maybe it was a ChIP-seq experiment, or a GWAS. Let's make a Venn diagram. I'm sure you've all seen these things, and you make that Venn diagram, and the things in the middle are now supposed to be the things that are real because they showed up in both data sets. Okay? Make that Venn diagram over and over and over again at every single different FDR cutoff that you can think of for either sets, and now you'll see that that thing gets bigger and smaller and goes away and comes back. And again, why is the Venn diagram intersection at 10% FDR somehow special? It's not. It's just completely arbitrary threshold. So we need a better way to find overlaps, right? To find really what we mean is not just that the same thing happened, but that their responses or that their differentiation was correlated across those different experiments, right? If something is differentially expressed in this experiment and it's going up when you give it a drug, and in some other experiment, some other entity is going up because you give it the drug, then that means that they're correlated, right? So we should be able to use statistics to be able to identify those correlations directly and not rely on this kind of post hoc Venn diagram method. Okay. So correlation networks, that's what we want to get to. So those were the numerical problems with the bottom-up approach. Then there's the human problems, the sort of arbitrary fickleness of any given scientist with a set of data, right? And this is actually one of the projects. We're going to give the same data to a couple of different people. They're going to use a couple of different pathway tools, and we're going to see what they come up with. We're going to see what they actually say they think is going on. And there's going to be different interpretations, right? Or even if there's the same sort of hypotheses that come out of it, they might be given different strengths or different rankings, right? And again, if I give you a genomics data set and then force you to tell me what is the hypothesis you're going to test next, what is this, in the absence of all other things you know, what did this data help you to learn that you are now going to go test? And if you now ranked those hypotheses from the first one you would run off and do right away, and then the second one down to the fifth one, every single one of you would give me a different ranking, probably. Or some of you would have the same ranking, but it would be very diverse. Okay? And again, there's no right answer. We all have biases about what we expect, certain hypothesis. I've already talked about this. There's this kind of overinterpretation of the data that when RNA abundance goes up, that must mean that the promoter activity went up. Well, maybe it did and maybe it didn't. You didn't actually get to observe that, but we tend to put that into the mechanism directly. That happens in Western blots and phosphoblots and everything. We don't really have any statistics. If we had these 5 hypotheses that we could all consider, we could vote on them, and maybe we'd get a consensus majority vote on what the best hypothesis would be to happen next. But we don't have any statistics. We don't actually have any math to help us rank them or put a threshold on the likelihood of success or the sort of risk probability to go with testing any of these hypotheses given the data. So that means sometimes, to be frank, the follow-ups are supported by wishful thinking. I'd really like this to work, because it'll be really interesting if this works. Okay? And then conversely, I'm like, oh, geez, I don't know, it looks too risky. Let's go do something else. So pessimism, right? And I guess I'll just end my I guess now 30-minute diatribe on this topic. Did the genomics, which you spent a lot of money, a lot of time, a lot of effort on obtaining, generating, analyzing, understanding, did it lead you to something you weren't going to already do anyway? And I think there's a lot of labs that I've had experience with where the answer was no. They already had an idea where they were going to go. They already had an interest in some particular pathway or some process or single gene hypothesis. They did the genomics because they wanted to kind of explore what was going on, and oh look, those blue dots said that my gene of interest was doing something interesting. I'm going to go do that. Okay, well, you were going to go do that anyway. So, all right, enough diatribe. Onto what the tools are. Okay, so there are a wealth of different pathway biology tools out there. These are just a small few of them. They vary in terms of their popularity and in terms of their overall size. There are some very small databases that are incredibly useful, even though they're not very popular. There are some databases like KEGG and Reactome and Bind and DIP that EcoPsych, Phosphobase, RegulonDB, MetaPsych, these out here on the right, you may have actually heard of. Incredibly useful, very popular tools. They've been in the community for a long time and have a lot of take. Reactome, we'll talk about gene ontology. We'll talk about GSEA and the database called MSigDB that GSEA uses. The Ingenuity pathway analysis tool is a commercially available pathway analytics tool, available to academics for a mere $10,000 a year. But your bioinformatics core may have access to it. So that's something to explore. But with all of these, fundamentally, every one of these provides you coordinated lists of genes arranged either in kind of bags of genes, sets of genes, or as pathways, right? So KEGG gives you metabolic pathways organized as as the sort of Behringer-Mannheim wall chart, right, of metabolic pathways. Okay. So the first class of pathway enrichment that we're talking about is overrepresentation analysis. And essentially what we're going to do is we're going to make a table. And for each gene set that we get out of KEGG or Reactome or IPA or a Go term, a set of promoter elements that a transcription factor might bind to, a set of microRNA targets, a set of disease associations, it doesn't really matter where the list comes from. And this is a point that for any given gene set, you can think of it not just from a pathway biology perspective, but from a disease biology perspective, or a microRNA targeting, or there's any number of sources that cross multiple aspects of biology that you can think about saying, is there enrichment in those lists in my set of interest? Okay. So here the idea is essentially we're going to make a 2x2 contingency table, and we're going to say there were my genes that were differentially expressed at some arbitrary threshold, 10% FDR, 20% FDR, 2% FDR, whatever you like that day. I'm going to put all of those genes over in this bucket, and then everything else goes over here in the non-expressed. And then I'm going to say, can I further divide those two sets into those genes that were in the gene set of interest? So this may be the gene ontology term for kinases. Okay, so 800 some different kinases in human. Okay, so 800 of the kinases, and some of them are differentially expressed, some of them are not. And then a whole bunch of other genes, 24,000 minus 800 that were not in the gene set, and so that were either differentially expressed or not differentially expressed. So these four numbers here, and I apologize for the kind of geeky mathematical symbols here, but you have four numbers that you're in the set and you're differentially expressed. You're looking at how big does this number get relative to these other numbers. Okay, does that make sense? And from that, you can do a statistical test to test how these things look. So let's quickly talk about gene ontology. We've mentioned it a few times. Probably most of you have been exposed to it. The gene ontology is a list of essentially dictionary terms about biology. And then each term, not only is there a definition provided for the term, but also a relationship that these specific terms are examples of something else. So a catalytic activity is an example of a transferase activity. I'm sorry, this is running backwards. Sorry, yes. So a transferase activity is an example of a catalytic activity. And we can go over here. A glutathione transferase activity is an example of a transferase activity that transfers an alkyl or aryl other than ethyl groups, or sorry, other than methyl groups, which is a transferase, which is a catalytic, which is a molecular function, which is everything. Right? So these are the three main branches of the gene ontology: the biological process, cellular component, and molecular function. Okay? And so any given gene might be annotated as having glutathione transferase activity and being in the cytoplasm and being involved in a metabolic process. And in fact, Bill's favorite gene, glutathione transferase mu1 from human, has those annotations. It's annotated as being in the cytoplasm, it's annotated as glutathione transferase, and it's annotated as having to be involved in some metabolic process. And the thing you'll notice about gene ontology is that this is somewhat specific, glutathione transferase activity. This is kind of very general, cytoplasm. So it's a cytoplasmic protein. That and a whole lot of other stuff, okay? And it's involved in a metabolic process. Okay, whoop-de-doo. Not a whole, not very specific, but that's what we get. When we get to the gene ontology and look at this gene, these are the sorts of information that you get. But now you can imagine for each gene ontology term, for transferase activity, or for cytoplasm, or for metabolic process, or for some more specific term in metabolic process, we can take all of the genes that have that particular annotation and ask whether they are enriched in our gene set. Sorry, was there a question? No. Okay. One other thing to say about gene ontology is that when you take those annotations of whether a particular gene is or is not involved in one particular term, you can be more or less strict about where that annotation comes from. You can insist that there's actual some sort of experimental evidence. These are all sorts of different evidence codes that they give to each annotation. Or you can say, you know what, I'm willing also to take computational methods Or I can be very, very strict and say, I need a traceable author statement. This is one of the most strict terms that says, there's a paper somewhere, a peer-reviewed paper, that says that this enzyme does this. And it's been directly assayed. Okay? So there's direct evidence for it. Okay? So this gives you a number of different levels of scrutiny that you can make gene sets even more refined and more high quality, or more loosey-goosey and include things that may or may not be true, and allow you to have some different sensitivity and specificities. So here's that same table again. And again, you're asking, are my hundreds of candidates involved in some similar process, pathway, or function? And this is what we do, the hypergeometric test. Okay? You will not have to do this on the midterm exam, I promise you. But the tools that you use are going to be doing this. Okay? And again, we're just looking at, is this number here that is in the group and significant, is this significant to insignificant ratio, is that really any more than the ratio of significance outside of the group? Do we see enrichment here? Is there something surprising? Is there some surprise that the statistics here ask you, how would you have just gotten this just by random chance, just by randomly assigning a group? Okay? So when you do this with Ontology, and you do it at each and every different level, or each and every different Go term, you get results that tend to look like this. So this is from this tool called Gorilla, which I think you may get to use when we do the pathway workshop. And the idea is that this term came up significant, statistically significant, and so did this term, and so did this term, but that's because if there are genes that were annotated to this term, then by hierarchy, they are also these things above, right? So let's go back real quick to glutathione transferase. So glutathione transferase is annotated as having glutathione transferase activity, but that means by inheritance, it is also a gene that has transferase activity, because the Ontology tree says that anything that has a glutathione transferase activity also has just transferase activity, also has catalytic activity. So there's a bunch of other genes that these Go terms would have besides glutathione transferases that would go off in different directions in the tree.
It doesn't matter for anyone's scope. I understand how something significant or specific at one point will continue to be specific as you move into the next specificities. But if you go
right, sorry, it doesn't go the other way. If something gets annotated as having transferase activity, like that may be, I may know that this enzyme has a transferase activity of some sort. I don't know what it transfers. I don't know what its substrates are.
Sure.
So I could annotate that gene with this Go term. I could say there's an association between this Go term and that gene. I would only be able to go this way. I would not be able to go down. I would not say that that gene now is a glutathione transferase.
Oh, sure.
Yeah. So it's only one way.
Right. But you can see that for the middle branch, it actually kind of seems to go back for a little bit. Instead of component, it goes set. So it doesn't matter.
So the component.
My question is really, it doesn't matter. You're comparing specific terms individually, right? When you do these comparisons,
yeah.
It doesn't really matter what branch it's on, how many steps there were before.
It doesn't matter. We're gonna look at so in this initial way of doing it, we're gonna just do each one of these by themselves. But any gene that was annotated to this Go term and or any more specific child Go term, those genes are going to get included in the set. Okay? Which is why you get this kind of pattern where things that are close to each other tend to also be significant because they essentially contain the same genes. You're testing the same gene set over and over and over again. Because there's not a whole lot of genes that either come in or drop out as you go down the list. Okay? There are ways to correct for that. You would like to kind of say, well, where's the real action here, right? Is the action here and this is just dribble down, or is the action here and if I kind of pay attention to this set of genes and then subtract that set of genes from everything else, is there actually more residual activity somewhere else? And that's what this is. When you correct for multiple inheritance, you get pictures that look like this, where now you see that there's just a very small number of things going on, and all these other things now become less significant. So you get the exact processes, the more specific gene lists that are actually enriched in your terms. Okay. So expected versus observed numbers of DEGs, and so hundreds of tools for this, all over the place. Again, my favorite, Gorilla. There's the link. We'll try and use it. But you can do this at the NCBI website. And we'll talk about that when we get to the workshop. This is the simplest thing you can do, and you can do it with such a wide variety of databases and things is what makes it attractive. That's the kind of the utility of this, is that you can look for enrichment 1,000 different ways. Like you can say, are my genes enriched for what chromosome they're on? Are they enriched for overlap with other genes that change in other sorts of experiments that I might care about? Okay. So before we go on, we have to talk about a little bit of not so much statistics, but actually philosophy. When we do that overrepresentation analysis, we're doing a particular kind of test which is called competitive. Right? We looked at this table and we said, what is this number that's in the set and its ratio to the non-differentially expressed genes compared to the number of differentially expressed genes that were not in the set? And this is called a competitive test. Because we're asking about the genes and whether, and we have a null, so these are what I'm telling you here are the null hypotheses. These are the null hypotheses that we're trying to reject when we do these tests. And we're saying that the null hypothesis is that the genes that are in the set are as often differentially expressed as the genes that are outside of the set. So the C here is the complement. Okay, so genes are in the set, genes that are outside of the set. And we're saying that essentially this ratio, this frequency of differential expression is just as high as it is outside of the set. Okay? So that means it's competitive. We're competing. So if there are thousands of genes that are differentially expressed, thousands of genes that are going on, and there's lots of differentially expressed genes that are outside of your gene set, you can have a lot of differential expression in your gene set, and now it's not significant anymore because you've had to compete with all those other differentially expressed genes that are doing something else somewhere else. And that may be dissatisfying to you because you think, oh, well, but there just may be a bunch of different pathways and a bunch of different gene sets that are actually getting turned on. And so why should one gene set get penalized because some other gene set is also active? And so that's one of the problems with competitive tests, is that an abundant, robust response can actually squash the power of these things to find their results. A different kind of test, is what we're going to look at in a second, is a so-called self-contained test, which is only about the genes that are in the set. It's really only about these two numbers and not at all about any of the other numbers. Right? So that's self-contained. And there the null hypothesis is a very simple one that says no genes in this set are differentially expressed. And you're testing whether 1 or 2 or 15 are differentially expressed. So that sounds good. One of the issues is that in the self-contained test is you might have just one differentially expressed gene, one very strongly differentially expressed gene, and now the entire gene set is significant, which is not exactly what you were kind of looking for coordinated, regulated biology across the entire pathway, and now it's just the one gene that's kind of driving it. So a little give and take here. And I think I just said all of this, so we can move on. Testing for the presence of any differential expression. These are overall more stronger. So if there is 5 or 6 genes in a gene set that are all weakly differentially expressed by themselves, you don't see statistical significance. These are the tests, self-contained tests, that now allow you to say, even though I don't have gene level significance, the pathway on a whole is significant because I'm seeing some amount of differential expression. I don't know exactly where it is. I can't point to any one gene and say this is the gene that is differentially expressed. But the amount of signal across all of the genes in the set is sufficient to say that there is at least one gene that is differentially expressed somewhere, possibly more. Okay. So the issues with overrepresentation analysis: arbitrary significance. The magnitude and directionality of the differentially expressed gene is not taken into account. So you can have a DEG at 10% FDR that has a 2-fold fold change, one that has a 10-fold fold change, one that has a 100-fold fold change. It doesn't matter. It still just gets a count of 1. Okay? It's very sensitive to that kind of the genes that are not in the set? What is those other numbers on that second row? What's that, the universe from which we're counting? And it's sometimes not really clear how to come up with those numbers. Do you talk about all the genes in the human genome? Do you only talk, so in a microarray or SNP chip, do you only talk about the genes that were on the chip, the ones you could possibly measure? Do you talk just about the subset of genes that had sufficient signal that you could even possibly call them DEGs? So some kind of low expression filter that you said, well, these genes were so lowly expressed that they never had a chance to even be called DEGs, so I shouldn't penalize them for that. So there's some issues with the universe and some biases. When we talked about RNA sequencing, we talked about the fact that the more highly abundant genes have more precision from RNA-seq, and also the longer genes have more precision in RNA-seq. So more abundant and longer genes are just easier to call as differentially expressed, and therefore when we go back and calculate these numbers, in any given gene set, if a gene set is composed of short, low abundant genes, then it may not be fair to use the same criteria for that gene set as some other gene set that has more highly abundant, longer genes. So you like to correct for those biases somehow. Again here, correlation between genes is ignored. We're treating them independently, and there's issues with correlation and crosstalk between the pathways. So now we're going to move into talk about functional class scoring. Functional class scoring is a self-contained test, and it's going to fix problems 1 through 3. So we're really excited about that. And the number one functional class scoring algorithm you will ever hear about is something called GSEA, Gene Set Enrichment Analysis. It came out of the Broad Institute a number of years ago. And the idea is that we're not going to just make some arbitrary threshold of FDR 10% and then treat all of those as equally likely as to be in the genes that are not. We're going to rank the genes from top to bottom as to how statistically significant they are with respect to the differentiating the phenotype of interest. So what you're supposed to see here is that this gene at the top, these genes at the top are more red over here and more blue over here, and conversely at the bottom, these are more blue over here and more red over here. And in the middle, it's just noise. So the genes are ranked from top to bottom. In this case, they're ranked by their overall significance of their fold change from both the most highest fold change A over B to their lowest fold change A over B, or downregulated in B versus A. And then the question is, if I now look at any particular gene set, I want to ask, well, where do the genes in that gene set, where do they fall? Are they randomly distributed across the whole result set, or do they tend to cluster either at the top or the bottom or both? And that's essentially what GSEA tries to do. And what it does is it starts with the first gene, and then the second gene, and the third gene, and it goes on. And at each step, it says, at that point, how much enrichment do I see? Okay? And so as I walk down the gene list, if there is enrichment, then this red line here will start to kind of randomly bop up and down, and it will keep increasing and increasing until the enrichment kind of stops. Right? So the rest of the gene set is essentially randomly distributed, so now the random walk kind of drops back down to the bottom. There's no further enrichment. We call these things Virginia plots, and we like to see Virginia plots. We're from Virginia, and why did I say that? Oh, I said that because the thing that we care about is how high this particular peak went and how quickly it got there. Right? So if this thing kind of randomly walks over here and it doesn't get very high and then comes back down, that's not as good as a case where it screams up to the top here and then drops back down. Right? So the statistics now come on, are not at the level, there's no p-value or FDR threshold on the input. All these are just ranked. They're simply ranked. There's no threshold. These are all the genes in the entire set. But now we get statistics as to how extreme or how interesting this Virginia plot happened to look.
The random walk, every time you find, the way that plot is constructed is what exactly? Every time you find one of the lines?
Yeah, so right. So this is an enrichment score that says how much, so very much like the overrepresentation that we just looked at, how much excess is there in the set versus not in the set, having gone as far as I've gone so far. Okay, and it's something of a counting exercise where you get a +1 for every gene that's in the set and a -1 for every gene that's not in the set. Okay, normalized by the number of things you've already seen so far. So that's why it goes up and then it goes down. Okay.
So in the end, you get a number.
In the end, you get a couple of things. One is for each gene set that you search, you're going to get a p-value, the probability of this being random chance or not, a false discovery rate, given the fact that you didn't test just one set, you tested probably thousands of sets, pathways, or Go terms, or whatnot. You're also going to get what's called the leading edge subset. It's going to tell you what were the genes that actually led to this significance. What were the ones that were at the top, essentially? Now that's interesting because the leading edge subset, these Virginia plots can actually go pretty far down into the weeds here. And sometimes just by random chance, the ones at the bottom here will not really be very strongly predictive of your phenotype. We're not strongly associated with it, but the idea is the whole set is. But the leading edge are your kind of leading candidates for explaining why that set became significant.
Is there any significance to finding a set of genes that you might call a lagging edge? Downregulated?
Right. So in this case, these genes were, in this gene set, they were mostly upregulated. Good point. If I rank them in the way they're ranked here, where the upregulated are here, then downregulated here, I can also, GSEA will also detect, so there's sort of three cases. They're all upregulated, they're all downregulated, or they're both, right? Where you have a mixture of both upregulated and downregulated. And GSEA will detect all three of those cases. Yeah. Okay. So that was functional class scoring. Now we're going to sort of go to the next step. So we've been talking about set enrichment, but of course these genes oftentimes, not all the time, not for Go terms, but for Reactome and KEGG and other pathway databases, we actually have information about how those genes are oriented with respect to each other functionally. And we know that, how these, or at least we have an expectation, a prior knowledge expectation about how they relate to each other. And so there's a class of tools. Again, SPIA is the one we're going to talk the most about, but then other tools like it that try to do the set enrichment from the perspective of the topology, such that if the enrichment is happening here at a gene that is kind of peripheral, or if you think of this as a graph that has central hubs and then spokes and more kind of peripheral leaves, if the enrichment is happening at the leaves, then maybe that's not as interesting as enrichment that's happening at hubs or at upstream, more sort of causal, if you will, places. So that's what this tool does, SPIA. And SPIA gives you a very informative plot that tells you, here's the significance of the set, of the pathway with respect to just looking at it from a set point of view. Okay, and so we see that there's some pathways that are very significant with respect to the set overlap. And then here's the perspective of looking at what is the probability that this pathway is actually perturbed given the directionality and the significance and the topology of the pathway, given where those nodes are actually falling, where the genes are falling in your set. And here what you can see is that of the ones that have great set overlap significance, there are a few of them that also have perturbation significance. But these guys don't. And so now in terms of considering all of these pathways, I would say these pathways would now be more interesting because they actually seem to be involving a perturbation to this pathway as opposed to just an overlap in the pathway that may actually be more peripheral than central. The other thing you get to see are cases like this where on the set overlap perspective, they weren't so remarkable, but now on the perturbation perspective, or at least jointly between perturbation and enrichment, now this pathway becomes a little bit more interesting than it was before. It separates away from the noise. Okay. The problem with pathway analyses in general is that we think about each of these distinct pathways, but they're not distinct, of course. There's lots of crosstalk, lots of overlap. And that crosstalk and overlap, when you start talking about, MAP kinase may be the end of the chain in one particular pathway in the database, but is in the middle of the chain in some other pathway. Okay? So the folks that wrote SPIA, this is actually the SPIA result on the left for a particular data set. White adipose fat from mice fed a beta-3 adrenergic agonist, comparing day 3 to day 0. And the top-ranked pathways they get are Parkinson's disease, Alzheimer's disease, Huntington's disease, and leishmaniasis. It's hard to make inferences about these things from white fat from mice fed a beta-3 adrenergic agonist. Doesn't necessarily make sense. Okay? But if you use a method that corrects for crosstalk effects in this, now essentially you go to the right, where these things go away. These things were all being driven by pathway crosstalk, essentially mitochondrial activity, phagosome, PPAR gamma signaling pathway, crosstalk into these other pathways that are, these are true and relevant pathways in these disease states, but probably just not active in this particular system. So when you see pathway lists like this, there is a little bit of, back to what I started with, there is a little bit of scientific interpretation that has to happen. But it's usually easy to spot things that are just not relevant and are probably can probably be explained away by crosstalk. Okay. So that's great. We have all these pathway databases that tell us things that we already know, and we can look for biology that already exists, essentially textbook biology that already exists. Are those pathways and networks and gene sets showing up or present in your data? What about novelty? How do we find those black and orange nodes that can't be explained by the textbook, can't be explained by Reactome, right? And I've been saying all along, we have to take into account the correlation somehow. So one way to do it, one of many ways to do it, is this tool called WGCNA, Weighted Gene Co-Expression Network Analysis. And this was developed for microarrays, but it works for RNA, it works for ChIP-seq, it works for peak data. It works in any situation in which you can say, I've got a bunch of samples. Let's just start there. I've got a bunch of samples. They may be all the same, in terms of their, a bunch of different people, a bunch of different blood samples. They may be experimental samples in which you've, done some sort of perturbation, but you don't tell WCGNA about that. You don't tell it about the perturbation. You don't tell it about the experimental design. You just say, here's some data. Go to it. And what WCGNA says, it says, all right, well, I'm going to look at this particular gene over all the samples. And then I'm going to say, how does the variation in this gene over all the samples compare to the variation of this gene over all the samples, or this gene over all the samples? Is there correlation? Can we find correlations between genes over the samples? Now that variation may be just random variation, because if a gene is not actually being modulated or regulated in any way across those samples, then there's not going to be correlation because it's really just noise. But if some gene is going up or down, and some other gene is also going up or down coordinately regulated with it, either in the same direction or in the opposite direction, then WGCNA says, oh, look, those two things are correlated. Okay? So it now makes a matrix, a correlation matrix, and says, OK, for all the genes, here's the correlation I see between gene 6 and gene 1, gene 10 and gene 1, et cetera, et cetera. And given that correlation matrix, it can calculate a correlation network, right? So if I take this matrix and say anytime the correlation is above some threshold, and there's a little bit of tuning that goes into this, if there's a correlation above the threshold, I'm going to draw a line between those two genes. And if there's not, then I'm not going to draw a line. And what you end up getting are these co-expression networks now where you get strong connections between certain genes that are all correlated one to each other. And then loose connections between, between genes where there's a little bit more separation. And so what you can then do with these networks is say, Here's a module, here's a network module in which there's strong correlation between all the members that are in that part of the graph. And here's another module where there's strong connections for that graph. Then for each module, you can then go back and say, okay, does this module's co-expression, does this module's sort of average expression value, does that correlate with my experimental design? Does that correlate with my phenotype of interest? Okay? So now you're doing testing not on tens of thousands of genes, but maybe on 20 different modules. Okay? 20 modules of strongly correlated genes. And then you can further go and say, okay, well, what are those modules? You can take those modules back to the pathway databases and say, can I now look for enrichment using overlap to say, is this module here enriched for a particular Go term or a particular pathway? And sometimes you'll find that, oh, this is the MAP kinase signaling pathway, or this is downstream RAS activated genes. And now all of a sudden you have that textbook biological understanding of it, but you've included genes that maybe were never in the pathway in the first place, and now you've got some genes that now, oh look, these four genes are all part of the are downstream of RAS or are activated by RAS, and this is one of those black nodes that nobody ever knows anything about. Now I have a causal prediction about maybe this gene is regulated by RAS. I can go test that directly. All right, so this is a method to cross that bridge between novelty and then also to bring that novelty back into sort of textbook pathway biology. Yeah?
Take two genes, for example. You are assuming that they are being expressed at the same level. What I'm trying to say...
Not the same level, correlated. So one can be really low, one can be really high, and if they're both going up and down at the same extent, they're correlated. Good question. Okay. What is the tool that you use to visualize these things and look at them? The tool is Cytoscape. I think Lisa's going to be talking a little bit about Cytoscape. So this is my placeholder to say when you want to look at these networks and nodes and things, Cytoscape is your tool. And here's the recap. Genomics is discovery research. It's not hypothesis testing. You have to just kind of own up to that in the first place. It can be difficult to publish a paper that just has a genomics result and no further follow-up. It's definitely difficult to write a grant and get it funded if it's just genomics without any hypothesis testing. Okay? Manually reviewed gene lists, intuition and creativity, a great thing, but I prefer some form of automated hypothesis generation. But just remember, any evidence of enrichment is not going to be causality. Many, many different sources of biological information. Here's where you can use your intuition and creativity. Don't say, I'm going to go to Gorilla because Aaron Mackey told me to, and I put my gene set in and I didn't find anything. So now I'm done. There's so many resources out there. We've touched on some of them. Lisa's going to talk about some more of them. There's resources out there. This is where you can actually really have fun with your data because it's more about the biology and less about which command line flag do I need to get this or that. Okay? Always be aware of this issue of competitive versus self-contained testing and this kind of trade-off that in a competitive test, if there's lots of things going on, then you may have reduced power to be able to find things because you're essentially competing. Any one given set is competing against all the other stuff that's happening in the other sets. Conversely, in a self-contained hypothesis, you're going to get a lot more power, but you may get too much power. You may get too many results. One of the things that happens with GSEA is instead of getting 5 or 6 pathways, you might get 300 pathways that are significant. And now you have to think about how those are working. Okay? Questions? Great.